This is part two of the immune system. And I'll try to make some sense out of some of the things I talked about during the first part. That is, talk about some of the interactions between the cells of the immune system, the stimulation of some to divide and proliferate, and also talk about some parts of the immune system in different places in the body and how they react to antigens, and then sometimes respond inappropriately. As I indicated in the first video, it's a very complex system and set of reactions. Some can be very specific, and then sometimes things can go wrong. So first I want to talk about some of the specific cells and some of the specific things that those cells do and then talk about their interactions with other cells. So there are two general classes of these cells. The first are the myeloid cells, and I talked about this in regards to cancer and a little bit in the first video on the immune system. And so there are cells here, some involved in blood clotting, some in carrying oxygen, and then some in different responses such as vascularization, dilating the blood vessels, and causing local inflammation, fever, and swelling in some tissues. And then the monocytes down here are primarily for two reasons. One is this dendritic cell, which is an antigen-presenting cell that picks up the epitopes of the proteins from the invading organisms and then presents those to other cells. And the macrophages can immediately phagocytize certain types of cells, such as bacteria. In fact, they chase them around in some cases. And they also signal other cells, including being signaled by other cells, including the dendritic cells and some of these white cells up here, the basophils, neutrophils, eosinophils. They can also be signaled by T cells over here. So the T cells are the central part of acquired immunity in signaling other cells to become activated or inactivated, and also in signaling B cells to differentiate into plasma cells once the proper antibody has been essentially discovered by recombination, and then enhancing the number of those antibodies to attack the specific antigen on the invading organisms or proteins. Each one of the cell types has specific as well as general receptors on their cell surface, on sticking out of their membrane. And so this allows them to react to different signals, including interferons, interleukins, to tumor necrosis factors, as well as many others, and in general called cytokines. So the first cell I want to talk about are these dendritic cells, and they take on several forms, but here's how they look diagrammatically. But in the scanning electron microscope, you can see these many folds and extensions of the membranes. And so on those extensions, they have cell receptors that will attach to other cell receptors, specifically on macrophages, on T cells, on B cells, as well as attached to the antigens of the invading pathogens. And so they're quite versatile, and the extensions of all these membranes allow them to display many, many different receptors on their surfaces. So when they have these antigens, indicated in red here, attached through the major histocompatibility receptor through the T cell receptor on the T cell, these, in addition to other receptors on that connect the T cell with these dendritic cells, will then, depending on what the signals are being given through these, they will activate release of certain interleukins and interferons, tumor growth factors, etc., that will then stimulate other T cells, here Th1 and Th2 and other types of T cells, to produce different cytokines from each one of those. And what they release are specific for the specific type of signal of the conveyed by this dendritic cell to the T cell through those receptors. And in the first video, I talked about some of those receptors, and many of them then have their activity by stimulating through signal transduction the activation of transcription factors, which then act on specific genes to turn on expression of certain proteins and eventually products, so that this T cell here, the Th1, will produce primarily interferons and some other cytokines, so that that will increase cellular immunity, although it also will increase autoimmune responses, and will also help in intracellular pathogen clearance. For the Th2 cells, and I mentioned part of this in the first video, they produce primarily interleukins, which will stimulate other cells and aid in humoral immunity, but are also active in asthma and allergic reactions. It also helps with extracellular pathogen clearance as well. So for each one of these, there's a combination of cytokines that will stimulate each one of the cell types, and that will stimulate those to produce other cytokines, which will act in different functions of the immune system. But as you can see also, when they go wrong, they can cause other things to happen, including disease states. So as you can see, it's a highly regulated system, but also because it's killing cells and attaching to proteins, there has to be a great deal of control so that it doesn't affect the main organism and the cells of that organism, and sometimes it does tend to do that. One of the main interactions between receptors is this one, and it can be any cell here, but often it's the dendritic cell that has become an antigen-presenting cell. But many cells that become infected will put an epitope out on the MHC1 receptor, that is, an epitope from the pathogens that have infected that cell. And then a T cell receptor that is compatible with that. That is, it fits into the cleft perfectly, so it recognizes that epitope also. And then there's a CD8 receptor also that connects those all together. In CD4 cells, this would be a CD4 receptor. And as you can see here, this is a more detailed view. In the case of CD4 cells, then the antigen would be presented on an MHC class II protein, which has two sections to it rather than one for a class I molecule. So here on this side for the CD8 cell, here's the T cell receptor, and here's the MHC class I receptor, which is presenting that antigen. And if the connection is correct, then the CD8 receptors will attach to that and strengthen that connection. In the case of a CD4 cell, it's almost the same, except the two receptors are slightly different. The T cell receptor is the same, but the MHC class II receptor here, which is presenting the antigen, and the CD4 receptor, which attaches that and firms up that connection, differ for the two interactions. Anyway, once these finally join up together, that will signal the T cell to interact in some way or react in some way to change its level of expression of certain products, become differentiated into one of the several types of T cells, and then start functioning to produce other types of cytokines to react specifically to that antigen. However, it's not that simple. So here's the T cell receptor here, the antigen in the middle in red, and the MHC receptor here. But there are other receptors that have to link up in order to have that APC, the antigen presenting cell, stimulate the T cell. So it's very specific. Here you have an interferon, which is not only picked up by the T cell, and then that causes signal transduction to increase transcription in the T cell nucleus. But those interferons also stimulate the antigen presenting cell also on slightly different receptors. And then interleukins also do a similar thing, stimulating transcription. But then these other ones have to also be together to allow that T cell to become activated and differentiated. So it's very specific and very complex. It's also highly controlled, so depending on what receptors attach together, you can get a co-stimulatory activity. That is, the T cell can be stimulated to increase transcription, and the antigen presenting cell can also be stimulated, not only to stimulate itself, but it's actually stimulating other types of cells, both B cells and other types of white blood cells. But it's controlled in that you can also have a co-inhibitory response, so that even though there's an antigen presented there and they start connecting together, the signal is not given to stimulate the two cells. In fact, they are inhibited from reacting. So this keeps the immune system somewhat in check, so that you don't have any sort of autoimmune or allergic responses. Just as the immunoglobulin locus was fairly complex, the MHC locus is also fairly complex. MHC1 class is a bit more simple in that there are fewer parts, and you kind of saw that from the one of the slides before, where it was only one molecule rather than a double molecule. The MHC class 2 is more complex in that there are pieces of those, again, similar to in immunoglobulins, where different sections can be used for different MHC class molecules. There's also a class 3, which is even more complex and isn't shown in this particular slide. So T cells really are the central signalers in the immune system after they get a signal from an antigen presenting cell, such as a dendritic cell or infected cell. CD4 cells, those are the ones with the CD4 receptors on them, as well as a couple of other minor types, are natural regulatory T cells. That is, they're always in a regulatory mode in the middle of the immune system. And it's the CD4 receptor that attaches to HIV, by the way. So that's why these cells become infected with HIV rather than any of the other T cells. And then you have also naive CD8 and naive CD4 cells that can be induced to become regulatory T cells or these other two types of T cells. And these are then induced to regulate the immune system under certain circumstances. So these would be induced if a certain type of pathogen were infecting the body of that person or cells of that person. And the CD4s would just naturally do that most of the time, so they're more generalists. And here are some T cells also that are first naive, and then they're primed by getting a signal from an antigen presenting cell. And depending on what type of antigen is presented, these will then differentiate into several different types of T cells. And you can see by TBET and GATA STAT6, these are the transcription factors that then are induced in those cells, and those then turn on specific pathways for different cytokines, including interferons, tumor necrosis factors, interleukins, etc. And so these ones here will turn on interferon, tumor necrosis factor, etc., and they're primarily antiviral and antibacterial. These ones mainly interleukin, and these turn on immunity to extracellular parasites. And then transforming growth factor will regulate the immune system somewhat, so it will tolerate at least some cell types, and it will also cause some cells to proliferate. And then down here, these are responsible for inflammation and fungal immunity, and then these ones down here are for T cell helpers, and also for B cells. That is to stimulate proliferation of those cell types. So as I said, it gets complex in a hurry, and this just shows some of the interactions between these different cells once you have an antigen. So the antigen up here is taken up by the antigen presenting cell, in this case a dendritic cell. It then puts that out on the MHC receptor that interacts with the T cell receptor of these THO T cells. And also the antigen infects these viruses, in this case, infect this cell. That also becomes an antigen presenting cell and puts that protein out on an MHC type 1 that attaches to a cytotoxic T cell, T lymphocyte. In this case, this THO then stimulates a Th1 type cell that produces interleukins and interferons that stimulates this cytotoxic T lymphocyte to kill that cell that's been infected. On the other side over here, you have stimulation of the Th2 cell, which interacts now with a B cell that can recognize that same antigen because it's attached to the virus particle through its immunoglobulin that happens to recognize the epitope of the protein on that virus. That sends a signal through to this B cell, and the T cell recognizes that that's the same antigen that it's seen back here. And it stimulates now the B cell to do one of two things, and this is happening in the lymph node. So it causes proliferation of those B cells. Some of them become memory B cells. Others become plasma cells that produce and release large quantities of that antibody or immunoglobulin to kill that virus, which has that protein on it. Here the focus is for the acquired immunity, starting with the mature dendritic cell here, signaling the CD4 T cells through attachment of the MHC class 2 receptor, which is presenting the antigen there, and the T cell receptor. And on the other side here, communicating with the CD8 T cell through the MHC class 1 receptor, which is presenting the antigen there, through the T cell receptor into that CD8 T cell. So in the case of the CD4 cells, they will produce interleukins, and that will cause signaling to the Th2 or Th1 cells, depending on the interleukin. And those will produce either interleukins or interferons, and both of those together can stimulate B cells to become plasma cells to release the antibodies into the fluids of the body. And those B cells and their antibodies will be specific for that initial antigen. The stimulated CD8 T cell will secrete interleukins, and the Th1 cells over here, the CD4 type, will secrete interferon, and both of those together will signal other cells to come in and connect up with a virus-infected cell, including these natural killer cells, and those will essentially dissolve or disrupt the membrane and cause dissolution of those virus-infected cells. Because they're releasing a lot of viruses when they do that, the antibodies will already be in place there so that they will attach to the antigens of those viruses and contain those as well. Here is a little bit more detail on those natural killer cells and how those are activated and deactivated. Eventually you have to slow those down. So on the top you have a pathogen that's interacting with the dendritic cell, that's then releasing interferons and interleukins, and that stimulates the natural killer cells to maturation, as well as activation of their cytotoxic activity. Down here, there's a slightly different reaction where the dendritic cells are activating the CD4 and CD8 T cells through that MHC2 receptor presenting the antigen, and then connecting up with the T cell receptor and the CD4 receptor as well. And so those will activate these Th1 cells that will in turn activate the natural killer cells, and they'll become mature and start killing any infected cell that they come upon. Also, these will release other cytokines, some of which will activate these cytotoxic lymphocytes, which will cause them to proliferate and increase their cytotoxic activity. When those are activated, then these natural killer cells that act early in the infection are then destroyed, so they go through apoptosis. So you have still a lot of cells being activated and expressing cytotoxic activity, but there are control mechanisms so that the host cells that aren't infected are not killed. So there's this tight balance between the two. Here's another type of white blood cell. These are the eosinophils. These exist in many tissues and can cause inflammation, but they signal a lot of other cells through release of various cytokines. They usually increase during parasitic infections as well as cancer. However, normally they make up only a few percent of the total number of white blood cells. So they are only increased in certain conditions. So these are the receptors. They have a MHC class 2 that presents the antigen and couples with T cells and other types of cells. Here's where it couples with a T cell, and it causes proliferation of these T cells and cytokine production. Another reaction over here is recruitment of Th2 cells, which will start to signal other cells to destroy any of the antigens that are remaining. Here it primes B cells, that is, causes them to make more of their immunoglobulin, in this case immunoglobulin M. It also promotes survival of the plasma cells in bone marrow. It causes maturation of the dendritic cells through also release of certain cytokines. It causes mast cells to release the histamine that they have, causing swelling, and it also increases the survival of mast cells, signals macrophages to become activated and start attaching to invading organisms. And the macrophages are capable of phagocytizing viruses and bacteria and just antigens. And then they also signal neutrophils to produce those reactive oxygen species, which essentially oxidize the invading organisms and their proteins, their antigens. And it also causes increase of specific cytokines as well. Monocytes and their derivatives make up a very diverse set of cells, and they start from these embryonic type cells, including the monoblast and promonocyte, and then eventually in the peripheral bloodstream you have inflammatory monocytes, which of course increase the amount of blood in the area, and then resident monocytes also just sort of hanging out ready for any invading organism that they can attack. In different tissues then, these can differentiate into other types of cells, still things that can phagocytize invading organisms, especially viruses and bacteria. So there, in the bone, there's these called osteoclasts. In the central nervous system, they are microglial cells. In the lungs, they're alveolar macrophage. In the liver, Kupffer cells. In connective tissue, they have these histiocytes. And then in the spleen, several different types of macrophage exist, ready for release in case of an invasion, an infection. White pulp, red pulp, marginal zone, and metallophilic macrophages. So all of these have similar properties as far as the cell surface proteins, but they're all just ready to attach to and phagocytize any invading organism, as well as any to clean up any antigen antibody groupings that they can find. And by the way, these can move around quite rapidly. So I've seen videos of this before where the macrophage is actually following a bacterium until it catches up with it and then phagocytizes it. So they can sense when a bacterium is in the area, and they chase them down and phagocytize and dissolve them. They also can signal T cells through dendritic cells, and so they send a signal to the dendritic cells. The dendritic cells then, depending on the signal they're receiving, can signal different T cells. Here, Th2 cells. Here are cytotoxic lymphocytes, Th1 cells, and other types of T cells, as well as signaling other monocytes also to proliferate and become, competent essentially for searching for that particular antigen. So one other type of white blood cell, these are the basophils, and on the left-hand side, these are the elicitor molecules. These are the things that attach to the receptors on the basophils that elicit a reaction. And then on the right-hand side, these are all of the molecules that are either released or presented at the surface of those basophils. So over here you have some interleukins that there are receptors for those that elicit a response in the basophils, usually to activate them. Note that there are also some immunoglobulins, IgG, IgE, and IgD, that are attached to the antigen already. So these attach to the Fc receptors. And actually many cells of the immune system have these Fc receptors, and so they will attach that constant region of the antibody to those FC regions, and that's how they attach. And so those become antigen-presenting functions on those basophil surface proteins. That makes this an antigen-presenting cell. They also, over here on the MHC1 receptor down here on the bottom, those also can present an antigen. So you can have two ways of presenting the antigen. They will also tend to agglutinate different cells together by attaching the antigen or to one or more cells, one or more other cells. And then these can be cleaned up by the macrophages. Over here you have areas where they can release certain chemicals, cytokines primarily. So there's interleukin-6, interleukin-4, tumor necrosis factor also. They can also release histamines, as can the mast cells, so that will cause local inflammation. And there are other things. There are antimicrobial peptides that are formed here for certain microbes. Those are very effective at killing those organisms. And then they have these CD40, CD80 receptors, and that's part of the interaction between these cells and other cells such as T cells, dendritic cells, and macrophages. Next, I want to talk about some specific interactions within specific areas and tissues of the body to sort of bring some of these things together. So here's one example of an infection through a wound. So you have a wound here and the bacteria are entering. Initially, blood platelets are released from the bloodstream. Then that starts to clot the blood immediately. But mast cells get into the picture quite early, and they release chemicals that cause dilation of the blood vessels, and they also recruit other types of white blood cells to the site, including neutrophils. And neutrophils will release those reactive oxygen species, which will essentially oxidize and kill those bacteria immediately. They also are able to phagocytize the bacteria and then destroy them that way. At the same time, they'll signal macrophages to produce cytokines, to release cytokines, or produce and release cytokines. And they also can phagocytize the bacteria. The cytokines will initiate tissue repair, but they'll also signal other white blood cells to come to the area to take part in neutralizing or killing all of the bacteria or all the foreign invading cells. So when you have a wound, initially you have the blood clotting from these blood platelets that are also produced by some of the blood cells. And then the mast cells release that histamine that causes both redness in the area because it's dilating the blood vessels and heating of that area, mainly caused by the increased blood flow to the area. So that's why a wound will often feel warm to the touch. In the lung lining as well as elsewhere, you also have dendritic cells hanging out there, and they will take in part of the virus or bacterium, as well as cell fragments, and they'll present those through their MHC2 receptors to the CD4 T cells. The CD4 T cells will then signal other white blood cells to come to the area and to fight that viral invasion. They can also phagocytize the viruses in another way and present those through to different types of CD4 cells. So they can actually produce many different activations of different CD4 cells that will then interact with, in some cases, multiple species of viruses all at the same time. And there are usually multiple things happening at the same time. So in this case, a macrophage is reacting to a particular virus by phagocytizing it, presenting that viral antigen on its MHC2 protein. That then attaches to a helper T cell through its T cell receptor. And then that signals proliferation of other T cells, cytotoxic T cells, which have been signaled also by an infected cell, one of the infected epithelial cells, for instance, through its MHC1 protein, presenting that antigen to the cytotoxic T cell through its T cell receptor. You then get proliferation of those cytotoxic T cells, and those essentially dissolve the infected cell. And as I said, this happens fairly rapidly. So you can see these things connect up together, and then the one cell just sort of dissolves right before your eyes. And this is what happens when you're infected with influenza or SARS-CoV-2 as well. So the dendritic cells signal the CD4 and CD8 T cells. The CD4 activated cells then elicit chemicals that cause the CD8 cell to signal a cytotoxic cell, which then sits right next to or attaches to one of the infected human cells and kills it by secreting certain chemicals that cause the membrane to rupture in that cell. At the same time, the CD4 cells produce cytokines that attract other white blood cells to the area and stimulate them to phagocytize any debris that's left over. And also, they will signal B lymphocytes to mature into plasma cells here that will produce antibodies towards that antigen of the virus, and those will be released throughout the body to also attach and neutralize all of the antigens and all of the virus particles that are left over. But as you know, when you get the flu, this takes several days to first kick in to make sure that you have the right antibody, and then to neutralize the virus and kill it. So there is a time when the virus is infecting cells, lysing them, and so this is when you get the fever and chills, when a lot of those cells are being killed all at the same time, which releases both the antigens, the viruses, and a lot of toxins into your bloodstream, which is what makes you feel ill from time to time. Tumors can also induce an immune response, and so some tumors will form and then be completely eliminated by the immune system. Alternatively, antibodies and interferons can be produced and purified in laboratory settings and clinical settings, and then used to treat particular cancers as well. The doctors and clinicians have to be very careful with these molecules and cells because they can be very potent and actually can lead to death of the patient, which has happened in some cases. On the other hand, in some cases, these immune therapies using either interferons or antibodies against specific tumors, very specific tumors, which they can target, have been extremely successful in many cases as well. So this shows a bit of the localization of some of these reactions. So in the thymus, you have a T cell here, a CD4 or a CD8 T cell, connecting through the T cell receptor into a natural killer T cell here, which then is mobilized to the lungs where the infection is occurring. So you have the microbes entering an antigen-presenting cell, which is a dendritic cell in this case, interacting with the natural killer cells that stimulates the release of certain cytokines, which activates the macrophages and the neutrophils. The macrophages then phagocytize and kill the invading organisms, and the neutrophils release the reactive oxygen species that also oxidize those organisms. At the same time, the antigen presenting cell is stimulating the T cells to stimulate the B cells to release, to become plasma cells and release the antibodies, which will also attach to the antigens on those microbes. That part is happening on these little patches that are in your lungs. So as soon as you breathe in, you're breathing in and breathing out, but you're breathing in a large number of bacteria and a large number of viruses with each breath. And so you have these patches inside of your lungs where you have these immune cells that are just waiting to react with anything that comes through there that happens to be pathogenic and starts growing. And so you're very well protected in your lungs, and I'll talk about the gut lining a little later too. There also are blood pathogens, right? So this is one that is specific to malaria, that is a disease caused by Plasmodium, a Plasmodium species. And so they have these molecules that are called hemozoans, which attach to the heme molecules, because those are the things that the Plasmodium is after, those blood cells and the hemes in the blood cells. So these attach to the hemoglobin in the blood and are taken in by the Plasmodium, but they also make people very sick. So these hemozoan molecules or assemblages are released into the surrounding tissue around the blood vessels. And this causes a reaction by the immune cells to initiate inflammation. This local inflammation causes fever also, but this activates the T cells and the B cells to form antibodies against those hemozoans. And then the macrophages take up that hemozoan and begin degrading it, but also signal that there's more hemozoans coming through, so it activates other parts of the immune system. At the same time, the dendritic cells are attaching the hemozoan, internalizing that, and then activating different T cells, including natural killer cells, which will attach to the infected cells and will kill them. And so it also produces interleukins and tumor necrosis factors that cause more inflammation and fever. So overall, when patients have this disease, they have localized swelling, localized inflammation, and fever, chills, things like that with malaria. Eventually, the immune system can catch up with this and kill it, but a lot of times the Plasmodium changes the proteins on its cell surface. So as soon as the immune system has worked out the antigen-antibody reaction and starts to kill the Plasmodium, a certain number in that population have already changed some of their cell surface proteins, and so they can survive the attack by the immune system and then later cause more disease by infecting more and more cells. So in most cases with malaria, patients have initially fever, chills, and then they feel better, and then weeks or months later go through the same thing over again, and then weeks to months later again and again. And this is primarily because those plasmodia are able to change those antigens with each cycle of infection. So as I already mentioned, in the lungs, there are these little patches that contain concentrations of immune cells, and the same thing happens in the intestines, a little different systems, but these are called Peyer's patches. And so when there's an invading organism, they're taken in, dendritic cells interact and react with those. They signal B cells. The dendritic cells also travel down into the lymphatic system and then train B and T cells train the B and T cells to react to those specific antigens. And then plasma cells are then released through the lymphatic system to attack that same antigen. Macrophages also are concentrated in those areas, and they will phagocytize the bacteria or viruses as they come through as well. You also have patches on your mucous membrane. So this is mucosal immunity. There are certain cells that are in little duct-like regions that release interleukins primarily. And there are also in these areas dendritic cells with at least one extension going up through the epithelial cells that interact directly with the pathogens. These then can signal either T cells of one type or T cells of a different type, including natural killer T cells. And then those will stimulate production of these cytokines that are released both into those epithelial cells to stimulate those to react to the bacteria or viruses, whatever happens to be the antigen-containing organism. But they will also stimulate the epithelial cells to release other chemokines and cytokines, which stimulate the basophils, neutrophils, and eosinophils to proliferate, and especially for the neutrophils, they release those reactive oxygen species to kill those microbes as they enter through those mucosal membranes. And there are many macrophages in the same area which will phagocytize the bacteria and viruses as well. In addition, there are often plasma cells in the area that have been released through the lymphatic system that are able to produce the antibodies towards those antigens that are coming through or at least deposited on the mucosal membranes. And those then are released through those mucosal membranes and so attack the pathogens before they even are able to get through the first layer of cells. So if you add up your skin and the mucous membranes, the lung linings, as well as the intestinal linings, you're pretty well protected. I mean, almost immediately upon contact of any invading organism or any organism at all that's not like your cells, they are immediately attacked by the series of different chemicals, antibodies, as well as cells of the immune system. And as you've seen, these dendritic cells are a major part of this whole process of taking the antigens into those cells and then presenting them through their MHC1 class receptors and presenting that to T cells and other cells as well. So almost any cell can be an antigen presenting cell, but these dendritic cells are really the major antigen presenting cell type of cell in this immune system. When they interact with CD4 cells, they will cause that cell to produce and release interleukin-10 that can be picked up by the dendritic cell, and that actually suppresses its function. They also produce other interleukins and tumor necrosis factors at low level, and they can stimulate CD8 cells as well. So they have a central um, role in the initial activation and communication with these T cells. So their developmental pattern is this. You start in the bone marrow with this hemopoietic stem cell. There's differentiation into a dendritic cell. They take in the antigen and process that and put out tiny pieces of that on their MHC receptors. And then they have other receptors on them as well. So they're presenting the antigen here, but they have other receptors that have to attach to different types of cells in the immune system. And then through those interactions, they produce different types of interleukins and other cytokines to stimulate natural killer cells to proliferate and kill cells, B cells to proliferate of one type usually, and then of T cells of many different types. They'll stimulate each one of those based on what antigen they are presenting to each of those cells. And here's a little bit more about these neutrophils. They're formed in the bone marrow, of course. They migrate into different tissues. And in doing so, they, in an immune response, they release these reactive oxygen species as well as other, including cathepsin, which we talked about earlier, which is a protease. They also interact with macrophages to produce certain chemicals as well as phagocytize different proteins, bacteria, as well as cells that have been killed by this process that were infected. So they're also central to the immune system, especially in the intestines, mucus linings, and lungs. Okay, this last section, I just want to briefly go over a few of the disease states that involve the immune system. So the first disease I want to talk about is inflammatory bowel disease. So there are ulcers associated with this disease, and they've shown bacteria in here, but some have indicated that fungi are also involved in disease progression. And then often tumors result from these interactions with the pathogens. So in this case, the T cells become hyperactive and start forming a lot of different interleukins and tumor necrosis cytokines, as well as the dendritic cells and macrophages. And all of this is causing inflammation around those areas, bringing more immune cells to the area. And also the tumor necrosis factor is causing cell death, and the macrophages also are leading to cell death. So these ulcers can become quite enlarged in the colon of the patients. Later on, too, tumors can be formed in these areas due to continuous irritation and DNA damage. And again, interleukins and tumor necrosis factors can be involved in this. Different types of T cells are involved, fibroblasts, macrophage. Neutrophils, of course, are releasing reactive oxygen species to try to react to the antigens formed by those tumors. So often with this disease, and it may be a collection of diseases, antibiotics are given to patients. Sometimes antimicrobial and antifungal agents are given. And in many cases, this can alleviate some of the symptoms and some of the disease. But unless it's treated, then often tumorigenesis is a possibility. Here are some defects in the immune system, especially associated with lymphomas, and these are of B cells. So this membrane with the blue coloring, that's the membrane of the B cell. Remember when we were talking about B cells and recombination, there's a lot of recombination going on in these B cells, especially in some loci, to create those specific antibodies. But that increase in recombination also can lead to some mutations via chromosomal translocation and other defects. And remember, from that defect of the diffuse large B-cell lymphomas, a specific translocation caused most of the changes in those cells, and it was due to a big change in transcription of certain genes. So one thing you can see right away is this diffuse large B-cell lymphoma. The defect is found in several places in those cells. The second one here is follicular lymphoma, and that's more restricted to just a few changes in the cell surface receptors and the production of certain cytokines. The next one is a mediastinal large B-cell lymphoma here, and that's exhibited in a few areas here on the cell surface. The immune-privileged DLBCL. Immune privilege means it only occurs in a few places, and that's in the central nervous system and in the testes, and so that's more limited. And then Hodgkin's lymphoma, which exhibits many of these changes. So in this top one, there's a problem with CD58 receptor as well as the MHC1 receptor. And so that changes signaling recognition for the natural killer cells. In this next one over, there's a problem also with the MHC receptor and communication with the T cell receptor on the cytotoxic lymphocytes. Also, again, with the MH2 receptor, communication with the T cells is interrupted also. And these are all present in some cancers of the DLBCL type, as well as a few other tumors. Over here, there's a problem with communication also between the B cell and a cytotoxic the cytotoxic lymphocytes, in that these receptors, the PD-1 receptor, is blocked from communication. And then coming down here, there are more problems with receptors as well as things like transforming growth factors and these galectin molecules, which stimulate the T cells and the cytotoxic lymphocytes. And so those are then faulty. The communication and coordination of the immune system is upset. And then finally, over here, there are many cytokines that are produced in higher numbers that then stimulate the T cells and the macrophages, the white cells. So then among these cancers of the B cells, these lymphomas, it primarily upsets the balance of those cells. So in many cases, there's cell proliferation where there shouldn't be, and interaction with antigens, cytokines, and other molecules is aberrant. And here's a summary slide of a tumor cell on the top where you have different receptors that are usually not present anymore. So there's lack of communication with T cells. And on the left-hand side, then, this causes activation of NF-kappa, which is a transcription factor, increasing transcription. Interleukin-2, which stimulates certain cells. mTOR and BCL also are activated. So all of these cause aberrant interaction between the T cell and recognition of a tumor that's gone out of control. They also, over here, because there's a lack of signaling, cause a decreased proliferation of the T cells, decrease in cytokine production, and decrease in cytosolic function, which then leads to a lack of activity or less activity in the T cells that otherwise should be reacting towards that tumor to kill those cells. So this is why in some cases, so if a cell becomes aberrant, then the T cells or the different parts of the immune system will recognize that that cell is faulty and will send out cytotoxic cells to kill it. In this case, the tumor doesn't have some of those sites on it, and so it's not recognized by the immune system, including the T cell. And so it's sort of hiding out from the immune system, which is one reason then why those cells can proliferate and cause a tumor. On the other hand, sometimes the immune system can start overreacting to certain things. And so there's an initial sensitization phase that includes interaction of T cells, B cells, basophils, and mast cells to create this sensitization for particular antigens or allergens in this case. And so the mast cells then produce histamines and other compounds which will stimulate other cells such as eosinophils, neutrophils, smooth muscle, and nerve cells to act in ways that essentially they consider that they're being continuously attacked by that antigen. These reactions are also influenced by the antigen being presented by the dendritic cell to T cells, which will then cause proliferation of the mast cells and cause them to produce more histamines. So then you get an early reaction, so that is less extreme than future reactions. And then finally there's chronic interactions with those antigens and allergens, which causes those symptoms, usually including irritation, sneezing, swelling, fever, and sometimes shock eventually. And much of this is caused by the overproduction and release of histamines from the mast cells, but also release of interleukins, tumor necrosis factor, other cytokines upon exposure to the allergens. And this can lead to some changes in the blood cells, mucus production, swelling, etc. The interactions between the dendritic cells, or antigen-presenting cells, and cytotoxic cells, these natural killer cells, can be of two types. And actually during an allergic response, you can get quite a few cells being killed in that response also, which also causes more inflammation, itching, pain, etc. And so there can be a couple kinds of interactions here. One is where you have the bacterium or allergen is taken into the antigen-presenting cell, the dendritic cell, and then it presents that on its MHC receptor, which attaches to a T cell receptor, and so you get a strong signal there, which causes a weak signal here. So cytokines are released, but not as many. So that gives a sort of a weak allergic response in this case. Alternatively, instead of a protein, a lipid antigen can be presented at the cell surface, and that creates a weaker bond between the T cell receptor and the MHC receptor on the antigen presenting cell. That produces a very strong signal to the cytotoxic cells, so they proliferate more and they tend to kill more cells. So that can kill more cells and result in a more severe allergic response. Finally, I just wanted to say a few things about a few other diseases that are caused by faulty signaling and cytokine production. And these include asthma and allergies, which are due to faulty Th2 cells or faulty Th9 cells or Th17 cells. And then autoimmune disorders, which can be caused by faulty dendritic cells, Th1 cells, or Th17 cells, and then fibrosis of some types can be caused by faulty T regulation cells, Treg cells. So these are other things. I won't talk about these in detail, but again, you can imagine that sometimes the cell surface receptors are faulty, or their response to those receptors is altered so that they either increase or decrease the amount of cytokine that's produced. And then just to draw this back to something we talked about early in the semester, and this is HIV infection, which affects the CD4 lymphocytes, which again you've seen from these past two videos that these are central to the immune response. So you have a primary infection where the HIV particles increase in number drastically, and that reduces the number of CD4 cells because they're infected and killed, or at least disabled by the virus. They eventually kill much of the virus, but some of it integrates into the chromosome of the CD4 cells. There's an eventual increase in the number of CD4 cells, but not to the normal levels. And then over time, the viruses keep replicating and releasing more virus particles from those cells that have been taken over essentially by the virus. And that infects more and more of the CD4 cells, eventually leading to a point where the number of the CD4 T lymphocytes is so low that the immune system can't effectively react to new infections of different pathogens. And eventually that leads to these opportunistic diseases, including fungal, bacterial diseases that affect the lungs and different parts of the body, as well as some cancers. And those usually cause death of the patient because the CD4 T lymphocyte levels are just too low to respond to any of those invading pathogens. This also brings up the point that these organisms are attacking or being introduced into your bodies all the time, but your CD4 T lymphocytes, as well as other parts of the immune system, are continuously reacting to them and eliminating them so that you can survive. And so it just shows that even when one component of the immune system is degraded or disappears, it's a fairly serious condition to the human body and to other animals as well. Okay, so that's the end of part 2 of the immune system, and thanks for listening. I will talk to you sometime later.